I'm really honoured to have been given the WHPS Prize this year. It's such a massive compliment to my team that our colleagues think so highly of our work and took the time to nominate me for this. And it's especially exciting to be given a prize by SFAM, which emphasises applied microbiology work, because to realise that colleagues see the potential for real-world impact in the research that I and my team are doing is just phenomenal. The prize was given for work which I've led on reconstructing and testing historical infection remedies, which are found in medieval medical books. And this is a research area that really initially grew from some chance conversations and an interesting side project into a major theme in my lab, and to becoming part of a collaborative network which spans the sciences and the arts and humanities to look at the history of medicine and its potential applications today. So in this talk, I'd like to tell you a little about the science that we've been doing and the people involved in this work. So the ultimate reason that I ended up doing this research is that I'm a massive nerd, and in my spare time my main hobby is historical reenactment. So an interest in learning sword fighting and historical martial arts led to a much broader interest in the history of the early medieval period. And I started looking into early medieval medicine because I wanted, when I went to living history shows, to portray a literate person, and a literate woman especially. And so I decided it would be good to take on the character of a leech or a doctor at living history shows, and I started reading into some of the sources around this period. And what I'm going to tell you today is how this ultimately went to this year in 2021, helping to lead my first ever phase 1 clinical trial to see if we could assess the potential safety of a medieval antibiotic, in inverted commas, to see if it could be used to treat infected diabetic ulcers. This story really starts with chance conversations with key colleagues at the University of Nottingham a few years ago, Dr Christina Lee and Dr Erin Connolly. Now, Christina and Erin are interested in the history of medicine, the history of disease, and in looking at some of the sources, the manuscripts that were written about medicine and disease in the medieval period. And when we got talking, we started to think, did any of these remedies actually work? Could we pool our skills and our specialities and try to make some of these remedies for infection from these medieval books and test them in the lab to see if they actually did anything? Now, this happened when I was doing my last postdoc with Dr Steve Diggle at the University of Nottingham. And Steve traces his career in microbiology really to a childhood fascination with the Black Death. So he was super keen to get involved in this work and see if we could try out a medieval infection remedy in the lab. And the other postdoc in the group at the time was Dr Aled Roberts, who has a long-standing interest in the antimicrobial properties of honey, which has been used to treat all sorts of infections throughout history. So again, there was a really nice overlap between the interests of a colleague in antimicrobial natural products and this idea to try and test historical infection remedies. And this is the recipe we picked to look at first in the lab. It's from a 10th century Old English text known as Bald's Leech Book. So Bald is the name of the person who commissioned it, and Leech Book is Old English for medical book. A leech is a doctor. And the remedy outlined in purple is an eye salve. So it's a treatment which you put onto a lump in your eye, which in Old English is called a wen in this text, and we think that's potentially a stye, so an infection of an eyelash follicle, bacterial infection. And the ingredients in this eye salve are really intriguing to a microbiologist. We're told to take garlic and a second allium species, pound them together in a mortar and pestle, mix them with wine and bovine bile, put that whole mixture in a brass or a bronze pot for 9 days and nights, and then strain out the lumps and apply the liquid to the person's eye with a feather. Now, those ingredients are fascinating to a microbiologist. Allium species like garlic, onion leek, etc. are particularly good chemical factories for making antibacterial secondary metabolites, and they've been relatively well studied in the lab to those ends. And then you have wine, so you have alcohol, which could be extracting compounds from the vegetable material, or conveying some antimicrobial properties itself, potentially bringing over some of the interesting small molecules from the grapes. And then you have the cow's bile, and bile is really good at punching holes in biological membranes, so it can burst open bacteria and kill them. Then you put them in the brass or bronze pot, potentially picking up copper salts, So this looked like a sensible one to test in the lab. And as if that wasn't good enough backing to test this remedy, the last 3 words of this remedy that were written down about 1,000 years ago are "Sibetste Lechtum" in Old English, the best medicine. So how could we possibly not pick this remedy to make and test in the lab? So we made it. This is what it looks like at the end of that 9 days and straining out. Now, we weren't sure of the translation of that second allium species, which in Old English is Cropleach, so we made 2 versions. In one version, we made it with onion for that ingredient, and in one, we made it with leek. We didn't use a brass or a bronze pot, we used a sterile Durand bottle, and we added pieces of sterile cartridge brass to mimic the copper alloy pot. And then we filtered it by centrifuging and using filter paper rather than straining it through a cloth, as the old remedy suggested. The first interesting thing we noticed is that this mixture was self-sterilising. So any commensal bacteria on the garlic and the onion, or bacteria from our hands or the grocery shop we bought them from, didn't survive this 9-day period of being combined with all of the ingredients. So that was kind of intriguing. And then we did a really simple well diffusion assay to see if this mixture could kill bacteria growing on the surface of an agar plate, and it could. But that's not very exciting, because a lot of natural substances, particularly a lot of molecules from plants, can easily kill bacteria growing on a petri dish, on an agar plate, but these same molecules we know can't kill bacteria in real infections. So that kind of assay is setting quite a low bar for finding out if something can kill bacteria. One big reason why this activity on a petri dish often doesn't translate into the clinic is that when bacteria infect soft tissues, like when you get a sty in your eye, they form a biofilm. This is an electron microscope photograph of a biofilm of the bacterium Staphylococcus aureus, and you can see here a biofilm is a multicellular aggregate of individual bacteria all stuck together by this sticky mass of material. This is a sticky glue, an extracellular matrix of carbohydrate and protein that the bacteria secrete, and it helps them invade your tissues, because this sticky overcoat can keep your immune cells out, and it can keep a lot of antibiotics out too. And the transition from planktonic growth as free-swimming single cells to growth as a multicellular sticky biofilm cues some big changes in bacterial physiology that give the bacteria increased tolerance to a lot of antimicrobial compounds. So biofilm is one reason why bacteria like Staphylococcus aureus are such effective pathogens. Staph aureus is the most common cause of styes, which we think our remedy was written for. It's also a common cause of conjunctivitis and a whole range of really nasty chronic soft tissue infections, like diabetic ulcer infections, post-surgical wound infections. And biofilms are a big reason why lots of candidate antibacterial compounds fail to translate from lab research into clinical usage. Lots of molecules can kill planktonic bacteria, but they can't enter the biofilm matrix, or they can't kill bacteria once they've switched their metabolism to biofilm mode. So we want to go beyond a simple test on a petri dish when we're looking at this remedy. We made Staphylococcus aureus biofilms in the lab so we could set the bar really high for checking the activity of Bald's eye salve. And we did this by growing the bacteria in little plugs of collagen and serum that mimic your soft tissues, letting them form a biofilm. And then we dropped the eye salve, or individual ingredients from the eye salve, onto our model wound biofilms, left them for 24 hours, and then recovered the bacteria to see how many were left alive. And you can see in our first results, which were very surprising at the time, that this mixture absolutely nukes mature biofilms of Staph aureus. Here's control-treated biofilms. We repeated this experiment twice, so you have individual repeats of the experiment from 2 different experiments in different symbols. And we can go from a healthy biofilm, 10 to the 8, 10 to the 9 live bacteria, to when we make our eye salve with either onion or leek, just a few hundred or a few thousand left alive. You can see all the round symbols from our first ever experiment are all overlapping, and that's because we actually set the detection threshold too high for this experiment. We weren't expecting to see this much activity. But the really interesting thing is you don't see this killing with any individual ingredient in the eye salve. If we use just one of the ingredients at the same concentration with the same preparation method, you don't get any activity. We also made versions of the eye salve where we had all of the ingredients but one, and those don't have full activity either. And we were actually able to show that you need the garlic, the second allium species, the bile and the wine for this to work. You don't actually need the copper salts from the copper pot, which was interesting, but you do need all of the other ingredients. So that was data from the first 2 batches we made and tested. And since that first paper, which was published in 2015, I and some collaborators got a project grant from Diabetes UK to explore the eye cell further. And our postdoc on that grant, Dr Blessing Anonye, who's now a lecturer at UC Lan, started to look beyond Staphylococcus aureus to see the spectrum of activity of the eye cells. And so here's just some sample experiments from what Blessing found using the synthetic lung model. So she's shown again, she can reproduce what we did the first time, and she can kill Staphylococcus aureus Newman. Here's data from 3 different batches of the eye cell. They're all quite effective. 2 of those batches are really completely knocking out that biofilm. Blessing also found she could completely kill biofilms of Acinetobacter baumannii, and as many of you will know, that's a really troublesome, really antibiotic-resistant wound pathogen that causes an awful lot of troublesome soft tissue infections. But Blessing couldn't kill any bacteria when it was grown as a biofilm in this wound model. So here you can just see example data from E. cloacae showing that you get really variable killing activity and often no killing at all. And that result, that we couldn't just kill anything, was actually quite heartening, because that's suggesting to us that whatever we've made has some kind of specific mechanism for killing bacteria. It's not some kind of general cytotoxic agent like bleach, which would not be very useful to take forward and investigate further. And various people in my lab have all made different batches of the eye cell and tested it, and we've shown that there's some really nice repeatability when you repeatedly make the eye cell from fresh ingredients each time. So basically, whenever someone new comes to the lab, we get them to make a batch of eye cell, test it on Staphylococcus aureus Newman. And of 84 batches that have been made in the lab, 71 of them, we see a greater than 3 log kill of the Staphylococcus aureus biofilms. And actually almost half of those batches, we see a more than 6 log kill of the biofilm. So this is really quite powerful in this model. And we've repeated that work on the individual ingredients and the versions of the eye cell missing one ingredient to confirm that you really do need all of the ingredients to make something that's active. This leads on to the next piece of research I'd like to show you. My PhD student Jessica Ferner-Pardo started to really dissect this need for all of the ingredients to see if the antibiofilm activity relies on more than one molecule present in the mixture. This is a picture of Jess giving a Zoom demo of making and testing the eye cell for an outreach event we did during the pandemic when we couldn't do outreach in person. And what Jess has first started to look at was the potential importance of the compound allicin in the eye cell. Now, allicin is a sulfur compound from garlic. It's been very well studied for its ability to kill bacteria in test tubes and petri dishes. And actually, we find if we test the eye cell just in planktonic culture, the presence of allicin in it explains most of its bactericidal activity. But allicin is not a useful drug candidate. It's got a very short half-life at physiological temperature. It's very reactive, has lots of different effects on biological cells, so it's not really something you'd want to use to develop into a drug. But what Jess showed was that allicin cannot explain the activity of Bald's eye cell when you grow it as a biofilm in the synthetic chronic wound. So on the top graph, you can see Jess has tested 5 different batches of the eye cells that she's made. They all completely eliminate mature biofilms of Staph aureus Newman in the chronic wound model, but Jess quantified the amount of allicin in each of those batches and showed it was quite variable. And then on the bottom graph, she's taken purified allicin at similar concentrations, and she's shown that at the top end of the range of allicin you find in the eye cell, if you add that purified to a biofilm of Staph aureus Newman, it's really not doing much killing at all. So something else in the eye cell is crucial for this antibacterial activity. So Jess has now been working with collaborators in the School of Chemistry at Warwick and the School of Pharmacy in Nottingham to work out what these active molecules are, because this will help us create a semi-synthetic version of the eye cell, which contains only the necessary bioactive molecules for antibiofilm activity. And this is going to be really crucial, because if we're thinking that we might be able to develop this cocktail of natural products into something with real clinical potential, we can't tell people, okay, when you have a wound infection, mash up some garlic and onion and add it with wine and add it with bile and stick that on yourself. That's not a good route to go. What we actually need is a defined mixture of compounds that can be reproducibly manufactured and delivered at known doses in a formulated antimicrobial agent or wound dressing. And this work is ongoing. But I'd like to switch tack now and come back to the idea of could a mixture derived from Bold's eye salve really have clinical potential? And to work this out, we had to move on to looking at whether it worked on a real biofilm in real animal host tissue, because a biofilm in the synthetic wound model is quite different from a biofilm in infected animal tissue. And also, our work on the synthetic wound model doesn't tell us anything about potential toxicity to animal cells or any unwanted side effects like promoting a huge inflammatory immune response. So this is where our collaborator Dr Kendra Rumbaugh and her team, particularly Rebecca GABRILSKA at Texas Tech University, come in. And what we wanted to do with Kendra and Rebecca was to see if the eye salve worked in animal tissue. So Kendra's lab have this really high validity mouse model of a chronic non-healing wound. And what they did was they took mice that had a wound infection with methicillin-resistant Staph aureus, then took a biopsy of tissue from this wound and exposed it to the eye salve, to saline as a negative control, or to vancomycin, which is the current gold standard antibiotic for MRSA wound infections. And what you can see, this is data from quite a short exposure window, just 4 hours exposure to the treatment agent. And you can see in this time, the eye salve is killing about 90% of the MRSA present in the biofilm in the mouse tissue, whereas in the same time period, vancomycin is only killing about 50% of the bacteria. So this was really quite something when we saw these data. And this then led to the question, is it safe? It might actually work in animal tissue, but would you actually want to put it on yourself? Would it be really cytotoxic? Would it be really pro-inflammatory? So we did 2 key experiments here. The first was a collaboration with Professor Laurie Snyder and her team at Kingston University, London. And what Laurie was able to do was work with a BCOP assay, a bovine corneal opacity and permeability assay, which is an industry standard model for irritation. And what you do here is you take cow's eyes from cows that have been killed for meat, and those eyes will actually respond to irritants. So you can see how much irritant potential a test agent has. And you can see here cow's eyes that have been treated with saline as a non-irritant negative control, and there's no damage to the cornea. If you treat with an irritant like sodium hydroxide, you can see this very clear patch of irritation. But with 3 batches of the eye salve, we saw either no or very, very minor irritation, which resolved within several minutes. To back this up, we then worked again with Kendra Rumbaugh and looked back at her mouse wound model. And here, without infecting the mice, we simply looked at whether adding eye salve to the wound had any effect on the normal healing of that wound. And you can see here from 3 batches of the eye salve, those wounds heal normally when the eye salve's added. So we don't have a huge inflammatory response, we don't see any necrosis. The animals didn't get stressed or start to scratch or bite at their wounds. So it didn't seem to have any negative impact on the normal wound healing process. So putting all these results together now so far, we're at the point where we got super excited about the potential of this mixture. And actually we applied for ethical permission to conduct a phase 1 safety trial. So this is co-led with Professor Julie Bruce, who's in the clinical trials unit at the University of Warwick, and my new postdoc, Dr Blessing OYEDEMI, who joined us a few months ago on the Diabetes UK grant. We were initially wanting to look at topical application of the eye salve or something derived from it, because while our ultimate goal is to test a natural product cocktail derived from the remedy on wound infections like diabetic ulcers before we can start to test on people with skin conditions or infections, sensible starting point is to check that the eye salve doesn't trigger any adverse effects when it's applied to healthy skin. Because if we see anything worrying when we put the eye salve on healthy skin, we definitely wouldn't want to test it on clinically vulnerable people. So we recruited 109 participants aged from 18 to 79, and we asked them to wear a sticking plaster impregnated with the eye salve for 2 days and then report any adverse effects to us. So the trial has now finished and the manuscript has been submitted to a journal, so hopefully you'll see the results very soon. What I can say now is that we're pretty happy that the eye salve is unlikely to trigger any serious adverse effects. So we're now at the end of our grant from Diabetes UK in the really exciting position of talking to wound care companies about translational research to really pull out cocktails of natural products derived from the eye salve and see if we can formulate these into advanced wound dressings and test them for their efficacy and safety in treating wound infections. You'll notice I've segued from talking about a remedy for an eye infection in the medieval text to talking about wound infections. Now, the reason for that is a lot of the same bacteria which cause unpleasant infections in your eyes are also the culprits of really nasty, non-healing chronic wound infections. And these are a huge health and economic burden. So by chronic wound infections, I mean wound infections that will last for weeks, months, even years. And these are things like diabetic ulcer infections, venous leg ulcers, pressure sores. And you can see here from some of the stats I've pulled out, these affect a large number of people. And the consequences of these non-healing infections are really quite serious. If they don't resolve, then they can lead on to osteomyelitis and to sepsis, and sepsis can be fatal. And so people with these wounds are given lots of topical and systemic antibiotics, and also physical treatments like wound debridement, negative pressure therapy. But actually, a lot of these wounds don't respond to those treatments. There's really extensive and unpredictable antibiotic resistance in the biofilms in these wounds. And this is one reason why chronic wound infections are so expensive to treat. And if you add up all of these types of chronic wound infection, the annual cost just in the UK to the NHS is over £3 billion. So we desperately need new agents to prevent and treat infections of these chronic wounds. It's really important, I think, at this stage to say you've got to be realistic when you're doing this kind of work. Most antibacterial compounds that look promising in the lab and in animal models ultimately fail to translate to clinical use for various reasons. But for this preparation, this medieval eye salve, to have cleared so many of the hurdles that we've put it through in testing and to have got to this point is just really promising and very, very exciting to be working on. Where else has this led? This has led beyond the work on the eye salve, so we've taken a much broader look at potential ancient biotics from historical medical books. So looking from things like these early medieval leech books right through to texts from the early days of surgery and the early modern period. And we've focused on wound remedies and soft tissue infections. We see an awful lot of mention in these texts of symptoms that suggest a soft tissue infection. So wounds that are hot or red, wounds that are foul and rotten, particular remedies for surgical wounds, dog bites, pustules in the eyes or the mouth. And we've been doing some quantitative research, seeing if we can turn some of these texts into databases for discovering combinations of natural products that we should bring into the lab for testing. And we've also done some qualitative research to look at how particular ingredients were used across time, across space, and for different symptoms. So we've been doing a lot of work with Christina Lee and Erin Connolly on looking at potential remedies involving nettles recently. But I want to come full circle to my nerdy excitement at working with medieval manuscripts. This is me with Bald's Leech book in the flesh at the British Library at their big Anglo-Saxon Kingdoms exhibition a couple of years ago, and they actually displayed it open on the page with the eye salve remedy, and the exhibition label cited our work. And that has genuinely been one of the most joyful and unexpected moments of my career to see this. So not only have we been able to study the science of this remedy and look at its effects on bacteria and see that there's something in there, there's something that we can start to dissect and potentially turn into a useful clinical agent, but we've also had this fantastic finding about the historical manuscript itself. I'd like to end this talk by acknowledging all of the colleagues who contributed to this work. So this interesting side project that Christina, Steve, Aled and I started in Nottingham 7 years ago was the seed for this huge collaborative network, the Ancientbiotics Consortium. And we combine history, data science, chemistry, microbiology and immunology to study pre-modern approaches to treating infectious disease. So thank you to all of the key people I've name-checked as we've gone along, and thank you to everybody else, from academic colleagues to undergraduate and MRes students, who've all made their own batches of iSalve during their research projects and contributed to our database of results. The early stages of this work relied on small amounts of funding from the University of Nottingham and Warwick, from the Microbiology Society, and from crowdfunding campaign. And since then, the Diabetes UK grant and Jess's PhD studentship from the MRC have been our critical sources of funding. And a huge, huge thank you to SFAM for their support of this work via the WHPS Prize.